Hello friends, welcome back to my YouTube channel. Today in this video we'll be going to see fourth semester previous year papers, that is 2024 and 2023. Okay, so here first we'll be going to see the 2024 paper, that is from September to October paper. Okay, so here you can able to see there was a five short questions asked here in the paper 2, that is cell communication and signaling name of the paper. So in this paper this was the question asked here, ionophores and gated channel. Do you know the ionophores examples is valinomycin, one of the thing which you have read in your first or second semester during this MSc, right? So you can able to write this answer easily. And MAPK pathway, this pathway I wrote in my notebooks with my handwritten notes. Those are very well written, diagrammatic based pathways are there. If you just learn for a while, you can able to understand the whole concept. Just you need to understand the diagram. Hope so I'll upload the videos of this pathways, or otherwise if you want to buy my notes you can buy it. Personally you can DM me if you want to buy the notes at the rate underscore, this is my ID of Telegram. So on that you can DM me so that you will get the notes. Okay, you can buy it. Now third question is cell surface receptor in plants. Fourth is functions of cytoskeleton. Cytoskeleton includes lots of things, myosin, actin, microtubule, everything included in it. Most of the things included in it, not everything. So you will need to write all the functions in under the cytoskeleton. The fifth thing is his kinase, is about the his kinase enzyme you need to write. Okay, and this sixth one, the question they asked in the alternate choices, first is cell cycle regulation. You need to write about the cell cycle regulation, checkpoints of the cell cycle, and also CDK2, like the cycle independent kinase enzymes. You need to write everything in detail about it. Now in this second one was asked about different molecule of extracellular matrix and the functions. You need to write about their functions. And then the seventh one is tyrosine kinase receptor mediated cell signaling process. So this is also a pathway, diagrammatic based. Another one was secondary messenger and the roles in cell signal. You need to write a different secondary messengers, that is calcium calmodulin and camp kinases, okay, PKC. So these are all the secondary messengers. Now eighth one was various mechanism adopted by plants towards the abiotic stress. So you need to write the mechanism of abiotic stress, various mechanisms. And here the next is plant signaling system with examples. So this is the paper of 2024. Now let's see the other paper. Here you can see this is a paper 1, biostatics and bioinformatives, name 2024 paper. So in this the short questions was asked about the central tendency. There you need to write about the central tendency regarding question answer. What actually the central tendency? You need to write about the details about mean, mode and median event. Okay, and NCBI, you need to write about what NCBI is. You need to write about the full form. And then third one is Sanger's method of sequencing. So what is the Sanger method and see how the protein sequencing occurs in it? That you need to write chi-square test and paleogenomics and applications of it. So these are the question asked in the short one. And if you talk about the long, here you can see bivariate data, about bivariate data, and along with that how regression is used to calculate the molecular weight of protein. So प्रोटीन का molecular weight आपको calculate करना था along with the answer of bivariate data. You have to write total about it. And here in the next question it was asked about the normal, binomial and Poisson's distribution. If you read this answer, that will be benefitable Okay. And other thing here was asked about the biological database, DNA sequencing database, then protein sequencing database. About that, you need to write. And the pairwise and multiple sequence alignment, you have to write. Next generation sequencing methods and HUPO goal and accomplishment. So this is the paper of your biostatics and bioinformatives from the fourth semester. Now let us see the other paper, that is paper 4, biotechnology paper. This is the 2023 paper. Here you can see what the question asks: microbial mining in the short, tissue plasminogen activator, and GM foods and social concerns, plant bodies, dolly, gene knockouts, pegylated interferons, drug delivery method. So these are the SAQs asked during the 2023. Here LAQs, if we talk about LAQs, production of human insulin and interferons, superbugs and its application. So in 2024, superbugs was asked as a SAQ. Okay, what are superbugs and about application? That's it. This one was question asked in a SAQ. Okay, then next is principle and application of gene slicing technique, plant tissue culture media, and immunotoxins tools for the cancer ट्रीटमेंट. काइमेरिक एंटीबॉडी के बारे से पूछा हुआ था. प्रोटीन टैग्स भी SAQ में पूछा गया था 2024 में. ओके, एंड सेल इमोबिलाइजेशन मेथड्स एंड इंडस्ट्रियल एप्लीकेशन, ये भी एक LAQ 2024 में रिपीट किया गया है. सो ये सारी चीज़ें जो है पूछी गई थी पेपर 4 में से. अब हम दूसरा पेपर देखेंगे. ये हमारा दूसरा पेपर 2023 का सेल जंक्शन सिग्नल ट्रांजैक्शन का. इसमें पूछा था साइटोस्केलेटन, जो कि रिपीट किया गया था 2024 में भी. सेल साइकिल रेगुलेशन को पूछा गया था LAQ में. हाँ, एंड फॉस्फोआइनोसिटेज इन्वॉल्व इन सेल सिग्नलिंग, कैम पाथवे, फिक्स लॉ पूछा गया था. एंड प्रोटीन इंटरैक्शन के मेथड्स, बैक्टीरियल प्रोटीन काइनेस पूछा था. यहाँ बैक्टीरियल प्रोटीन काइनेस पूछा था, वहाँ पर SAQ में हिस काइनेसिस पूछा था. ठीक है. एंड देन हियर अकाउंट ऑफ डिफरेंट ट्रांसपोर्ट मैकेनिज्म्स अक्रॉस सेल मेम्ब्रेन पूछा था. यहाँ एक्टिन, मायोसिन एंड द रोल्स इन ECM पूछा गया. जी प्रोटीन के बारे से पूछा गया था. सेकेंडरी मैसेंजर्स अगेन, ये तो 2024 में सेम टू सेम रिपीट हुआ है. सेकेंडरी मैसेंजर्स के सारे रोल्स लिखने हैं आपको वगैरह. सिग्नल ट्रांजैक्शन एंड सेल साइकिल रेगुलेशन, ओके. ट्यूमर सप्रेसर जीन के रोल्स लिखना था आपको. बायोस्टैटिक्स का सिग्नलिंग पूछा था वहाँ पर. यहाँ पर 2023 में और 2024 में बायोटिक का पूछा था. सो दिस इज़ द डिफरेंस. प्रीवियस ईयर पेपर्स पढ़ने से आपको एक आइडिया लगेगा कि कौन से आंसर्स आपको पढ़ना है. ठीक है. तो ये था पेपर 2 फोर्थ सेमेस्टर का. बायोस्टैटिक्स बायोइन्फॉर्मेटिव्स का भी पेपर है हमारे पास प्रीवियस 2023 का. तो आप यहाँ देख सकते हैं किस तरीके के क्वेश्चंस थे. बहुत ही टफेस्ट पेपर था. हियर यू कैन सी 4 ग्राफिकल रिप्रेजेंटेशन ऑफ डेटा, और फिर एक क्वेश्चन दिया था जिस पे प्रोबेबिलिटी देखना था आपको थर्ड बैग की. ये क्वेश्चन था व्हाट इज़ द डिफरेंस ऑफ पैरामेट्रिक एंड नॉन पैरामेट्रिक टेस्ट का डिफरेंस लिखना था. एंड फिर इसके बाद आपको पूछा सिग्निफिकेंस मीन्स इन स्टैटिस्टिक, रिसर्च शुड अप्लाई कंपेयरिंग टू कंडीशन, कंट्रोल एंड टेस्ट के बारे से. सीक्वेंस अलाइनमेंट के बारे से पूछा था. हायरार्किकल सीक्वेंस एंड शॉटगन सीक्वेंसिंग मेथड, 2D जेल्स, LC एंड MS इन प्रोटीन बायोलॉजी. कंपैरिजन पूछा हुआ है किसका, सीक्वेंस का एंड स्ट्रक्चरल कंपैरिजन पूछा हुआ है. सेंट्रल टेंडेंसी को LAQ में पूछा था, बट वहाँ पर SAQ में पूछ लिया. डिप्रेशन स्टैटिक्स एनोवा पूछा है LAQ में. क्वालिटी कंट्रोल इन बायोकेमिस्ट्री पूछा हुआ है. नेक्स्ट जनरेशन सीक्वेंसिंग 2024 में भी पूछा हुआ है LAQ में. पैम एंड ब्लॉसम का पूछा हुआ है. एबेंटो प्रेडिक्शन, होमोलॉजी मॉडलिंग एंड फोल्डिंग प्रोटीन पूछा हुआ है. पोस्ट ट्रांसलेशन मॉडिफिकेशन इन प्रोटीन. दीज आर द क्वेश्चंस आस्क्ड इन द 2023. So here comes our fourth semester completed previous year paper we have seen. And if you want more previous papers like this, do subscribe my channel and share my channel with your friends. And if you are new on my channel, please subscribe my channel. Thanks for watching.